Okay, well, let's go ahead and get started. I think we've got a good crowd here. Once again, my name is Brooks Bonner. I'm the Director of Community Engagement and Enrollment Management for the Organization for Tropical Studies. And I am excited that today we have a great presentation for you. But as you should know, and as you probably do know, this is Organization for Tropical Studies TropTalks, Stories from the Field, our online science webinar series. And today's talk is going to be about hemoparasites in Kruger National Park, and it's going to be given by our very own Tino Pori, who is a faculty member with OTS, who actually got his start with OTS as a TA in some of the global health summer courses. And while he was getting his master's, has completed his master's and is now taking on a larger role with OTS as a faculty member in not only the African Ecology and Conservation semester program, but our newly approved online course, Disease Ecology and Wildlife Management, which will be offered this summer. So I'm super pumped to have Tino here. And I was telling him earlier, it's kind of a rite of passage for a lot of our faculty in South Africa that they've got to stay up until midnight or 1 a.m. to give one of these talks to make sure it's at a decent time for all us people in the US. So thanks again, Tino, for doing this. And before we do get started, I want to make sure that people know that at the end, when this is done, there will be an opportunity to ask questions. And we hope that you would ask questions of Tino. And I think without further ado, I will pass the mic to Tino. Tino, you want to go ahead and get started?
Okay. Thank you very much, Brooks, for that introduction. Hello, everyone, and thank you very much for having me. I see a lot of former OTS students. Good to see you guys. Probably let me begin by giving a little bit of some context into this work. This is actually work that was begun by my former supervisor, Dr. Mduduzi Ndlovu, who was also a former faculty member at OTS. So I first got introduced to this work when I joined OTS as a student in the semester program. This was in fall 2015. And so this was one of the deliverables that I chose to work on. And I must say, I was really excited about the work. And I remember just a few days in, I was so convinced that this is the kind of work that I wanted to do. And like any other OTS student, I began to try and find ways in which I could actually come back and start contributing to understanding the ecology of diseases in the Kruger National Park. And so I did this work with OTS I came back and did this work with OTS for my master's, and then joined OTS as faculty. And then we continue to do work that is related to understanding diseases in protected areas. So as Brooks has mentioned, that the talk for tonight is entitled Avian Hemoparasite Prevalence in Kruger National Park, South Africa. So there has been a growing interest in the study of avian diseases following the rise in the emergence and spread of infectious and zoonotic diseases in recent years. But one may even begin to wonder, why is it important to study diseases in birds? Now, birds have a widespread geographic abundance, being found in virtually almost every environment. And the diversity among species of birds is also quite enormous. I mean, birds account for one of the most diverse vertebrates on this planet. But interestingly, is that avian blood parasites are correspondingly diverse. It is actually estimated that there are as much blood parasites as there are species of birds, which actually shows the great pathogenic potential of this group of hosts. And so as a result, therefore, now birds have been regarded as an excellent model for studying any host parasite interactions in the wild. But the prevalence of avian hemoparasite infections has been poorly studied and is virtually unknown. the determinants and the impacts of these infections on their hosts is quite difficult to estimate, particularly in areas of Southern Africa where infections are endemic and the host and parasites have managed to co-evolve. But though infections are of little clinical significance, they may, however, become pathogenic in naive hosts or whenever there are any changes to the ecology of the host, the parasite, or the vector. So this is basically a typical host-parasite interaction relationship in the wild, where it is not the intention of a parasite to kill its host, because that would be foolish of the parasite, as it would not have any way to feed from if it were to kill its host. So there's always a mutual a relationship between the two. And as you can see, that this is more like a seesaw kind of relationship where there's always a maintenance of a balance. But whenever there are changes in this kind of setup, either to the host, the parasite, or the vector, we then have threats of infectious diseases in an environment. And typical examples of where this has occurred is, for instance, in the Hawaiian Islands, where you find that there was the accidental introduction of Plasmodium relictum and one of its vectors into the environment. And this caused avian malaria to birds and mass extinction of birds in the Hawaiian Islands. And we've also had similar cases in South Africa, where some penguins that have been put into captivity have also been introduced to altered disease dynamics and have led to avian malaria in these birds in captivity. And one may also wonder how these birds actually get infected by these parasites. So infection by parasites is mainly through diphtherian vectors such as mosquitoes or different flies that we find in the environment. And they tend to take advantage of the eye area of the bird, or also in areas that do not have much feathers, such as the breast of the birds. And this often infects these birds and leaves them at risk of falling ill. And then some of the objectives of this study were to explore the spatio-temporal drivers of parasite prevalence. We wanted to find out if there were any seasonal variations to infection, or whether infections varied in different environments. And we were also interested in assessing the host intrinsic traits, and some of the traits that we're particularly interested in were the traits that are associated to the host's feeding guild. So we would have expected birds that actually forage closer to water bodies to have a higher diversity of parasites, because this is basically where the vectors breed, the mosquitoes breed close to water, and birds spend a significant amount of their time feeding. So you would expect birds that that forage closer to water bodies to have a higher prevalence. And by the spatio-temporal drivers, we actually expected to find seasonal variations in infection, because vector ecology is mainly driven by season, as vectors are dependent on temperature and rainfall. So you'd expect a higher prevalence of infection during the wet season, where you have high temperatures and you have a lot of rainfall, which is actually a good breeding ground for a lot of these vectors responsible for the transmission of diseases. And then another host intrinsic trait that we wanted to find out was the social status of the bird, to find out if birds that are gregarious are more prone to infection than those that are actually solitary. And you would expect birds that are gregarious to actually have a higher prevalence as they congregate in larger numbers, and the risk of transmission is quite high in such cases. And then we also intended to find out host immune response to infection, and by so doing, we wanted to see how infections affect body condition and how the body responds to infections through analyzing what we call white blood cell counts, which are a measure of testing the immune response to an infection. And then for those familiar with the Kruger National Park, this study was conducted in the Kruger National Park in 6 sites. It is important to note that there is a rainfall gradient that decreases from about 600 millimeters of rain per annum in the southern region of the park to about 350 towards the north. So there's a reduction in rainfall. So you'd expect a higher diversity of parasites or in the southern region where there is actually more rainfall. And then the sites, 4 of the sites were within the Kruger National Park, and the other 4 sites were at adjacent human settlements that are outside. The birds were sampled using mist nets and walking traps, and these are some of the students that were involved in collecting some of this this fieldwork. We were involved in this fieldwork, and this is Melissa untangling a bird in a mist net. This is Alex, and this is Jackson. And I'm always interested in understanding a bird's activity in trying to reduce the burden of diseases. And the bird handled here by Jackson, the crested barbet, is actually one such bird that plays that kind of role. So if you look at the crested barbet, it nests and roosts within tree cavities, and it always makes sure that it removes the fecal matter from the cavity quite often. And this is actually a way in which it reduces infections to itself and its chicks. And then, so birds were collected using mist nets. Blood was drawn from these birds, and in order to make slides and to collect blood for molecular work. And as you can see here, morphometric measurements were taken in order to determine the body condition score. And this work also involved several hours in the laboratory under a microscope, identifying parasites. And probably another interesting fact about this bird is the marabou stork, which is handled by Jackson right here in the middle where my caser is. So this is the marabou stork. You realize that this bird has a bare face, and it is actually, and it's quite a tough structure because this bird is a scavenger and has to dig through dead bodies, and then this structure prevents it from getting any blisters and cuts, which could lead to infections. And also, this bird also plays a very important role as it scavenges, because it goes for dead bodies, and it acts also as a cleanup in the environment, as it gets rid of several harmful bacteria and other infections in the environment. And then moving on to some of the key findings that we found out in this work. We got an average prevalence rate of 27% that was recorded from 7 genera of blood parasites. And this was after sampling 687 birds from 87 different species. And one of the key findings was that we did not find any significant difference between a prevalence across different sites. So prevalence was the same across different sites, which actually shows that these parasites are ubiquitous and are found throughout the Kruger National Park. But what is important to note is that we found a higher diversity of parasites in communities that are outside the park, which actually shows how anthropogenic changes play a role in increasing the burden of diseases through habitat disruption and also creating microclimates for some of these vectors. And then we also did not find any seasonal variations as we had assumed. And this could be attributed to the fact that probably the birds carry these infections for a period that actually exceeds the season in which they were infected. And another reason could actually be that during the time where we sampled most of our birds, we actually had a prolonged dry season. I mean a prolonged wet season, which could have actually altered the vector ecology and sort of extended the breeding periods for vectors to continue infecting these birds beyond the seasons in which we usually get the most infections. And also another reason could be that we are not even yet sure about all the vectors that are responsible for transmitting these diseases. Probably these vectors, some of the vectors have nothing to do with any seasonal variations. And then moving on to the host intrinsic traits, we also didn't find any difference with infection, difference in infection amongst different host intrinsic traits, such as the social status and foraging guild. So birds that foraged closer to water bodies were infected similarly to birds that forage on the ground or aerial feeders or canopy feeders. It was all the same. And also birds that are gregarious also had similar infection rates to the birds that are solitary. So moving on to the immune response, there was also no effect on the body condition. The infections did not cause any negative effects on the body condition. But in few cases, we actually found that infected birds actually had a much better body condition than the uninfected birds, which is quite unusual. But we begin to think that some of these birds have evolved and have learned to actually co-evolve and live with these parasites without them causing any effects on the birds. And then the white blood cell counts are a measure of immune response. We also did not find birds that are infected responding by having an elevated white blood cell count. And then these are some of the images of how these parasites actually look like under a microscope. This is the normal red blood cell here on my caser, and then this is the plasma around. So this is an example of leukocytes. This is how it looks.
Tino, I think your microphone is just rubbing a little bit there. Just.
Oh, okay.
That's better. That's better. Thanks.
Okay. And then this is leukocytosome, and then this is Plasmodium. So similar to how Plasmodium affects a human by causing human malaria, this is what actually happens also in a bird, where this parasite gets into the nucleus, it gets into the cell and tries to displace this nucleus off the cell, and then eventually cause diseases such as anemia. And then here you have hemoproteus, and then this is a trypanosoma, and then this is microfilaria, and then here you have hepatosome. So these are some of the parasites that we identified. And then in conclusion, these results actually reveal some complex relationships between parasites and their hosts within an Afrotropical environment. And there is further research that is actually required for us to understand the dynamics of diseases in the environment, particularly research aimed at understanding the ecology of the vectors and to find out how these function in different savanna management practices. And that actually brings me to the current work that we're now doing, be it with OTS and with the newly introduced disease ecology and wildlife management course. This is the kind of work that we're going to be doing. And so if there are any of you who are interested in doing this work, you better sign up for this work, because part of the themes of this work is to try and understand the complexities associated with human, animal, and environmental health at human-wildlife interfaces. So this program is going to be looking at that. And in disease ecology, when we try to address these disease dynamics, we sample for either the potential hosts or the potential vector. And in this case, we're going to be sampling for both. We're going to be sampling rodents for potential diseases. We're also going to be sampling for birds. And as potential hosts, we're going to be sampling for mosquitoes and also sampling for ticks to find out what parasites and burden of disease they carry, and to see how these infections differ at various land use practices within the environment. And thank you for your attention, and I'll take any questions that you may have.
Thank you, Tino. That was great. That was great. I want to open it up, as Tino said, I wanted to open up to questions. And I do think first, as people are gathering up the courage to ask their questions, I did want to make a note. So Anne had mentioned about explaining an illustration, but I'm not sure which slide that specifically referred to. Anne, did that slide get described for you, or?
Yeah, I think it did. It was right at the beginning.
Okay,
but
great.
Then it did later.
Okay, great. But yeah, as Tino mentioned, the disease ecology and wildlife management is an online course for undergraduates. That's going to be something that we offer this summer. And it's a new course. Obviously, it's still online. There's still limitations about travel, but that's a great opportunity to actually get some great experience handling, organizing, and analyzing data. So we hope that some people will be interested in taking a course like that. And it is for credit too, from the University of Connecticut. But yeah, I want to open it up now to questions for anyone out there. Apologize for the background noise. I'm getting some heavy rain here. No tornadoes, which is good. So Tino, maybe I'll start then. You talked about your interest in doing this type of work. Had you always had an interest or a curiosity in birds, or how did that come about?
I mean, in birds, it was not in birds, but I've always been interested in diseases, in understanding how diseases function in an ecosystem. So I remember growing up as a young boy. Each time I would visit my grandfather's farm, I always had like my favorite cows. And one morning, one of my favorite cows had actually died, and I was so sad. And when a vet doctor came to assess the animal, he said the animal had actually died of foot and mouth, which is caused by a buffalo, which is the host reservoir for this infection. But at that time, I had never seen a buffalo before. So I became curious to understand how this infection came about. And then, so from that time, I'd always been interested in trying to understand how diseases function. It was not necessarily in birds, but just diseases in general, yeah. So that's how my interest actually came about. But after coming to OTS, then it was actually now gotten bigger.
Great. Very cool. Well, I think someone does have a question. If, like I said, if you also, let me clarify, if you have a question, feel free to unmute yourself and ask away. No need to raise your hand, just go ahead and blurt them out.
Hi, I have a question.
Sure.
Can you hear me?
Yes.
Yes.
Perfect. Thank you for your interesting talk on the genera of parasites. I really enjoyed that. You mentioned right at the start that your research site is in close proximity to village communities by the Kruger Park. I'm interested to know whether there's a relation between that genera of parasites with local diseases that are emerging as a result of that. Do you see a correlation of local diseases as a result of the parasites people are surrounded by in the birds?
Okay, thank you. That's a good question. We sampled birds that outside the park, but then currently we haven't had any cases of any disease spillover of infections that we have found in our birds. So we haven't recorded any disease spillover, but perhaps probably if we had sampled for avian influenza, I know communities outside have had outbreaks of avian influenza, bird flu, in their domestic chickens, and they could have. We know that our ducks and wild birds are reservoirs for these diseases. So there's actually a possibility that actually could have come from areas such as these. But with regards to the work that we did not find any relation to poultry diseases that are prevalent in communities outside the park.
Okay, thank you.
Tino, thank you so much for this presentation. It's been so interesting. And one thing I want to let everybody know about, we've been starting a new initiative. We've had some preliminary and National Science Foundation funding to start up a network for tropical researchers to create their own online learning modules based on their own research. And I'll put it in the chat. Let's see, let me do it now. We've found an online authoring tool, so it makes it really easy to create your own online module based on your own research. And so we have an incubator grant. We've just been doing it kind of small time, but we work with OTS and ATBC. Those people are on our steering committee. We're trying to get the word out to tropical ecologists that it's a really user-friendly platform to just like cut and paste the things that you have for a learning module. And it seems like you have the makings for one. So check out Learn Gala and see if you want to make a module. and the people who created this software they really love our group, because everybody knows tropical ecologists are the most fun people on the planet, right? And so and it's funny because so we got this incubator grant to get us started, and then the idea was to submit a full proposal, which we did. We got declined. I just resubmitted a new full proposal. But honestly, the train has left the station. I mean, we don't have the full funding, but people are just going ahead and creating modules. And if anybody wants the links to these kind of draft modules, I'm happy to send them to you. I should put my email in the chat. But Tino, I really encourage you to create a module, it's really easy to do. That's what we love about the software. They make it easy.
Yeah, that's great. Yeah, I think, and Tino obviously is going to be playing a big role in that disease ecology and wildlife management course, which is online. And so that's a course that Tino is having a major role in the development and design of that course. So, and obviously within the larger full semester program, as you can see from many of the photos, students are very involved in, data collection and analysis and just kind of developing those field research skills, which is so much a pillar of what OTS is about. So yeah, thanks for sharing that, Ann.
That's the kind of thing that will make it so cool. Including those photographs of the students will make it so cool, because this NSF project we have, it's in the undergraduate biology education part of their education thing. So it's aimed at undergraduates. So this is
Great.
like perfect.
Nice. That's great.
And really the idea is to counteract, like in South Africa, you may not have this problem, but I'm in Iowa, and most of these kids, they've never even thought about going to the tropics. They don't get how cool it is and how much fun it could be. And so that's the world I want to open up to them, that, you don't have to be stuck in Iowa.
Absolutely. Absolutely. South Africa and Costa Rica are very different from Iowa. That's for sure.
And also, I don't know what your textbooks are like, but one thing that we've come to realize in our network is even in the tropics, the textbooks are based on the temperate zone. And what we really want to do, I think our ultimate goal is to rewrite the textbooks to put the tropical stuff into the textbook. So anything that moves us towards that is great. So I'll get off my soapbox, but
that's all right. Thanks for sharing, Ann.
Thank you.
Any other questions out there? I know we got some OTS alumni, or maybe if there's a few OTS alumni, they can share their experiences with maybe helping Tino along the way. I see Jessica and Emily are here.
Tino, thanks for a great talk. We're wondering, what are you most excited about for your next steps with this project?
So the next steps with this project are actually the work that we are now doing in this disease ecology practicum that we're starting. I am now mostly interested in understanding the vector ecology to address this challenge more vigorously, if I may say. So we intend to sample for potential vectors. I'm mostly interested in the mosquitoes, which are actually responsible for transmitting bulk of these parasites. So we're going to be trapping mosquitoes in different management systems. You are aware that the Kruger has got experimental herbivore plus and Kuhlu exclosures, which eliminate herbivores of different sizes. So we are going to be looking at how the species distribution actually varies in these different environments. And we also want to look at how these species also differ with different land use practices. So looking at how anthropogenic changes outside the Kruger National Park affect the dynamics of diseases by affecting the vector ecology. You are aware that there's a lot of land use changes outside. So that is also likely to have an impact on the burden of diseases. So I'm mostly interested in understanding what these vectors are carrying, what is it they are feeding on, and the whole relationship between the vector and its potential host. Yeah, so that's the next step. So you better find your next flight to South Africa and be a part of this.
That sounds awesome. Great to see you.
Good to see you guys.
It's always nice to hear some from some alumni. That's great. We're always, I know Lawrence is the king of Lawrence Kruger, the director of South Africa, is always, I think every time I talk to him, he's talking about he's trying to convince me to come to South Africa, or anytime he starts up a conversation with anyone, if we're at a college or university, it's always trying to convince them to come to South Africa. So the tourism board must pay him something. Okay, well, I do want to be conscious of the time. If there are any, I think we might have time for one more question. If there's any out there. But I do want to say, Tino, thank you again so much for sharing your work and giving this presentation today. This TropTalks is still somewhat of a new initiative, and we hope that we will, monthly, we'll continue to have these types of talks as a way for OTS to just share kind of the work that we do with the broader community. And I think, who knows, in the future, there may be some other opportunities for OTS alumni or any other OTS related folks that want to give a short presentation on the work that they do and how OTS has shaped that work. We're more than willing to listen to those potential opportunities. So you can contact me, Brooks Bonner at tropicalstudies.org. You can also go to the TropTalks page. And if you haven't already, please sign up for, if you go to the TropTalks page, there's a form at the bottom called Stay in the Loop. If you want to get notified on upcoming talks, that's the best way to do it. We'll send out a link and information about a talk that's upcoming every month or so. So please do that if you haven't already. And Tino, thank you again. And get some sleep. I think it's getting close to 2 a.m. in South Africa. So I think you've earned a good night's sleep.
Thank you. Thank you very much for having me.
Great. Thanks so much for everyone for joining us, and we'll see you next time.